So today we're going to do nervous system. I'm going to do a double lecture since we have a double class period if we have time. So that's going to be rough. I'll take a break in between the two lectures and you can go to the bathroom or whatnot. And I normally wouldn't want to take up that much time, but we just don't have much time left. So outside of class, you guys have the AP daily videos you can be watching, as well as practice questions within the AP Essentials class. You have the live reviews, which are now no longer live. Last week was the last of that. They cover all of the units. There are playlists that you can watch, AP playlists, like in the Google World, I have a playlist for review or a playlist for nervous system, a playlist just for AP year. So I'm posting a nervous system playlist today. And then AP classroom, I already said that one. And review books. Aren't you guys using review books? Because like they are awesome. They're kind of like cliff notes of each of the topics. And that's really zeroing in on what the AP world wants you to know. And then they have practice questions for each of the topics. And then they have practice tests overall. Their practice tests probably are not as wordy as the tests that you're going to see next Friday. Can you believe it's next Friday? And you'll probably miss class because you have other AP tests coming up. Any questions about like the next week, 2 weeks? So in your book, when you first read from this chapter, the first thing it did was talk about the evolution of the brain through the phyla. So the peripheral, the Cnidaria, the Planaria, the Annelids, the Mollusks, and the Vertebrates, these are really arranged from least to most complex. So you can assume evolution has added adaptations over time that have created more complexity. So I want you to compare these 6 phyla. Look at like the organization of the nervous system. Do they have cells? Do they have tissues? Do they have an organ system? What is the arrangement of the nervous system or arrangement of the body? So there's like bilaterally symmetrical, radially symmetrical, asymmetrical. What else? Just maybe what new adaptations have occurred in this phyla. Okay, so talk about cells, tissues, organs, new features that have arisen over time. Good. So they're multicellular. Do they have nerve cells? Do they have neurons? No. I see a head shake in the back. So no, they don't have neurons, which is what most of the other groups are going to have that allow them to communicate, but they do have cell-to-cell communication. So that's what we're getting into next, in that the release of different chemicals from a cell or different ions from a cell can cause a reaction in another cell. So we went from cells just communicating with chemical release to now we actually have nerve cells, which would include action potentials. And the nerve cells are all connected by a net, allowing them to talk to each other. And then we learned a little bit more about these organisms. They even have motor, sensory, and interneurons, which create a flow of information from the sensory to the interneuron, which is integration, and to the response, which is the motor. So here we have the coral, and the Hydra is so little. Do you see the little white guy there? Do you see the white thing right there? That's a Hydra. Oops. Anyways, so that's the Hydra and the coral, and they belong to the group Cnidaria. And then we don't have this one. This is Echinodermata. So Echinodermata means spiny skin. What kind of symmetry do you think this one has? Yes, they have radial symmetry. They would also have a nerve net, or I'm sorry, yeah, they would have a nerve net, but they have a nerve ring here and then radiations coming out from that. And then you talked about the ganglia, which is a cluster of nerve cell bodies. So then we go into our worms. So we have flatworms, roundworms, and segmented worms. The flatworm is the least complex, and that I have an example of a liver fluke. You guys know Planaria, maybe. If you were in Miss Cox room, you probably worked with Planaria. Okay, so now on to the Planaria, the flatworms. Flat.
Planaria have nerves that form ganglia, and they run the length of the body. They're bilaterally symmetrical. They don't have a brain, but they have primitive ears called auricles, and eye spots that are sensitive to light.
Good. So these are, when we say sensitive to light, we mean that light itself will cause a response, right? So action potentials occur in the presence of light. And then they'll either move away or toward. Remember, positive taxis and negative taxis. Okay. So bilaterally symmetrical, what does that mean? So the right and left halves. So like we are bilaterally symmetrical. The right mirrors the left. Good. And then we'll go to have our roundworms. This is an example of a roundworm, Ascaris. We're going to skip them. They'd be radially symmetrical, I guess. No, they'd still be bilateral, even though they're round. This is an earthworm. So you guys recognize earthworms. This is our segmented worm, Annelida. It's hard to focus through the water, I think. So tell me about Colin, can you tell me about the annelids? Oh my God, I never closed their rooms. I'm so dumb sometimes. Ay, ay, ay. Ay, 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 ay. Miss Snook is not smart today. Hi guys, we just been talking about the first three groups. Totally forgot to bring you back from your breakout rooms. So luckily we have shared these slides so you guys have all the information. We talked about the peripheral. They're the simplest. They have no nerve cells whatsoever. They do have cells that release chemicals that allow cell-to-cell communication. And then we talked about, I'm going to give you the coral because you can barely see the Hydra. The coral, which now has a nerve net. So nerve cells that are actually communicating with each other, but no organs. And then we talked about the Planaria, which was the flatworm. They're bilaterally symmetrical, meaning the right mirrors the left. And they have now a ladder-like nerve innervation. They have two nerve cords that are going from the anterior to the posterior. And then they have lateral nerve cords along the way with ganglia, which are groups of nerve cell bodies along the way. Now we're ready to talk about the segmented worms. So this is Annelida. Now that I got rid of my groups, I forget who was it. Colin, I think. Can you tell us about the annelids?
Yeah. So the annelids have a primitive compact brain in their anterior of their body, and they're connected with two central nerve cords that connect with the ganglia. And I looked into it, and it said their nervous systems have evolved by having that pair of ventral nerves conjoined to make one.
Good. Yeah, perfect. So we see we've gone from cells that are not neurons to cells to a tissue to now we're starting to get a brain. So now we're moving into an organ. So we can then go into the mollusks, which the good example is the squid and the octopus for this, but I'm going to give you I don't have them. So of course the clam, you guys are familiar with the clam. This one we also spent some time talking about symmetry. So you have asymmetrical like the sponge. You have radially symmetrical like the starfish, and you have bilaterally symmetrical like the earthworm. So two sides mirror each other. So this is a really good example of a bilaterally symmetrical because it has two halves, right? Two sides of the clam. So this is the phyla Mollusca, and that means soft body. We're going to zero in a little bit further on the cephalopods, which means head foot. You can be a gastropod. These guys are gastropods, stomach foot. And a cephalopod, which is a head foot. So that's your squid and octopus. And Declan, is this your group?
Yep. So mollusks have a tetraneural nervous system, which means there are 4 main neural strands. In mollusks, a pair of cerebral ganglia innervate the head, mouth, and associated sense organs. They're soft-bodied invertebrates, and they're symmetrical, and they have usually around 5 to 6 ganglia.
Good. And we talked about ganglia being clusters of nerve cell bodies. So we went from cells to tissues to organs, and now we've developed an organ system. And you can imagine the more complex the system is, right, the more communication can take place. And so now we're able to, like the brain is able to tell the body what to do. And then we have the vertebrates, which is you and me. Oh, I jumped fast. This is an amphibian, double life. That's a frog. We have vertebrates like the mouse. You can see the backbone of the vertebrate, perhaps, if he focuses. There, you can see the backbone along the purple along the back there. So that's a vertebrate, or you're probably not going to be able to see this one. The deer, this is a deer baby. It's actually a deer set of twins. Anywho, that's us. So we also have arthropods. I skipped to the arthropods, just my survey of the animal kingdom. It's a big giant grasshopper. Arthro means jointed, pod foot, so jointed foot is the arthropod. Okay, so tell me about the vertebrates, the most complex of them all. Josh.
You said that they were just like highly developed and that their brain controls their nervous system, and then they have like the special organs like the eyes, the ears, and the olfactory structures. And then they have like the most amount of neurons out of all of the invertebrates. They have like many more neurons, and they have a specialized nervous system. Their brain is broken, like can be like split into different kind of sections based off of size and function. And then we also, yeah.
Good. So now we have many different organs, which allows us to monitor our environment and respond to stimuli, right? So the purpose of the nervous system is to monitor both internal and external environments, respond to stimuli, and maintain homeostasis. So that's the job. So I'm gonna move this one down, and I'm gonna pull this one up. You are hanging out over here, which is not where I want you. Let's move you over here. So now I begin. So I wanted to give you guys a minute to do a little bit other than just listening. I have to change my screen, sorry. Okay. So this is part of the big idea number 4, which is interaction in systems. We should be able to compare the nervous system of the 6 phyla and for the most part, just identifying how we have evolved over time to go from cells to tissues to organ systems, and that complexity has occurred over time. I think I have to So hopefully you found that a little bit interesting. The survey of the kingdoms. So we want to be able to describe features of a typical nervous system within the vertebrates specifically. And then notice how the brain has changed over time, allowing us to become the most dominant species on the planet. And later, how the nervous system provides information, both input and output, as well as integration to keep the system working. So you guys just went through all of this. I think you did a fabulous job. We kind of talked along the way. So I don't feel like I need to go through this again. Is that good? You guys all produced this. I'm giving you this slide so that you can at least see the arrangement, if in case you didn't do, say, the Hydra, for example. So here you can see the nerve net with the cells all connected. And then radially symmetrical, you have a nerve ring. I was showing you that if you were in class, you got to look at the starfish. Here's our bilateral, 2 nerve cords with transverse or latted, like along the side to side. This has just the beginnings of the brain in the eye spot. And we can see the complexity occurring, more ganglia, allowing for more the 2 sides working together.
Oh, you're not sharing, by the way.
What's that?
You're not sharing.
Oh, thank you. Net, ring, bilateral. You guys will have these slides. I was holding them off until you guys did your section. And here you can see a ganglia at each section, right? And the vertebrates are like that as well. Each of your vertebrae, that begins a ganglia to go out to a specific part of your body. And we talked about the squid. The squid is considered the most intelligent of the invertebrates. Invertebrate meaning they don't have a backbone, vertebrates meaning they have a backbone. So we're going to spend a little bit more time now on the vertebrate nervous system. So in the vertebrates, we all have a hind, mid, and forebrain. Our hindbrain, back here, the green portion, this is the most ancient of our brains. So you can assume evolution has moved it forward to the forebrain, and the hindbrain is dealing with just the subconscious, like what's happening below the conscious level. So while you're sleeping, your heart is beating, you are breathing, all those things are happening without you thinking. So that's your hindbrain. The midbrain, more what was the first things that developed, right? We had the eyes and the ears, the auricles and the eye spots. So it makes sense then that this next section had to do with coordination of those responses. And then we get into the forebrain, which is where the thinking comes into play. So this is our conscious section. So this is the newest of them. The midbrain contains the optic, which is eye, and then auricle refers to the ears. And then the forebrain is processing sensory information, touch, taste, smell, sight. All of those things are happening here, processing, integration, making decisions. Paired sense receptors allow animals to gather information from the environment. So sensory neurons. Neurons are your transmitting cells, and the very end of them are your receptors. So you have photoreceptors responding to light, chemoreceptors responding to chemicals, baroreceptors responding to pressure. So whatever the first part of the word is, it tells you what it's responding to. Mechanoreceptors respond to mechanical movement, physical vibrations. So within the different vertebrates, the hindbrain and the midbrain are very similar. These 2 sections here, the forebrain is where the vertebrates, like each group of vertebrates, have a tendency to change. The outer layer is the neocortex. This is only present in the mammals. We are mammals. Other fur-covered, milk-producing, live-bearing animals are also mammals. This outer level is your higher level processing, your critical thinking, your decision making, your deliberateness, your ability to guess the future. that sort of thing. And so there's lots of complex connections that are occurring within the brain itself. The brain, if you look at the outer part, it's gray and the inner part is white. So the gray matter is more for the signal transmission. The white matter is where the cell bodies are. The cell bodies are where the action is taking place as far as the decision making, the production of chemicals, neurotransmitters that need to be released, different proteins, all that sort of thing. So you have gyrus and sulcus, these folds. We are the only groups that exhibit these folds. So the folds increase both surface area, which we've talked about. Increasing surface area makes for more chemical reactions, but also allows for more connections between neurons. So there can be more communication happening throughout the brain. And I don't know if you'll be able to see this. Again, things to a jar are hard to see. Oh, I don't feel like changing my screen again. Let's see. Okay, you can see the blue. That's the outer part of the brain. And then you can see the white, which is the inner part of the brain. So the inner cortex and the outer cortex. Okay. So the cerebrum, this portion here is the largest part of the brain. And this is where we are telling our body what to do, telling our organs to release different hormones or different secretions, telling our body to move. This is where we think. Everything physical, perhaps, and sensory related are happening in the cerebrum. Has a right and left divided by the cerebral hemisphere. Our brain. And as Sophia was telling us, each part of the brain is like it's able to divide this higher level, the vertebrates. We can divide our brain in different parts, do different jobs. Just like your cells, if different parts are doing different jobs, you're more efficient, right? So we're able to accomplish a lot more things. So this frontal lobe is your thinking portion. The parietal lobe back here, this is sensory reception. You have the temporal lobe, which is on the side. This is hearing. Occipital lobe in the back. This is for vision. You have the thalamus, which is on the inside here. It's a sensory relay, allows the midbrain and the hindbrain to communicate with the forebrain up top. The hypothalamus down here is associated with your drives, hunger, thirst, also associated with pituitary gland, releasing chemicals. Lots of different chemicals are involved, but one of them has to do with the reproductive system specifically. The cerebellum down here in the back, the little cauliflower looking piece, that is coordination and balance. Okay, that's my survey. Normally I pass those around, but I'm on this camera, which makes it a little different. The midbrain is just relay tracks connecting the spinal cord to the brain. The medulla oblongata regulates your heart rate. That's that hindbrain, the ancient part. So all organisms, all animals have this. And so all animals can regulate these things. Not all animals have hearts, obviously. And I shouldn't say all animals have it because as we learned, we didn't even get a brain until the roundworm. Okay, so that's the brain. Any questions on that? I have a video from anatomy where I talk about the anatomy of the brain and the pieces and parts, and I might go into a little bit more detail. So if you wanted to look at that later, you could do that. Now we're going to get into our nervous system. So this is the human nervous system. You have the central nervous system and the peripheral nervous system. The central nervous system is your brain and spinal cord. This is your integration. So the nervous system, and I've seen a FRQ on this. Remember, there's sensory input, there's integration, and there is motor output. So this is the middle portion of that. That is the integration piece. The peripheral nervous system is the sensory input and the motor output. So this is sending signals to and from the brain and spinal cord. So brain and spinal cord central, peripheral nervous system going to So this is according to the direction the nerve impulses are traveling related to the brain. Going to the brain is sensory. Going away from the brain is motor. So this is sensory input, decision-making, and then motor output. And if in your book, it refers to somatic and visceral. So somatic is referring to muscles, and visceral is smooth muscles, glands, and organs. Here we'll call it autonomic. But you have sensory information coming from both somatic and from the visceral, which is referring to organs. And then you have motor output going to both places also. So somatic is referring to muscle movement. So then you would get into like the contraction of actin and myosin. And then autonomic is referring to all of like your organs and glands and smooth muscles. Then we can get into the sympathetic, which is fight or flight. So this is for the most part increasing processes, not all processes. Some are going to decrease, like digestion is going to decrease. But sympathetic is you're either going to fight for your life or flee, right? So we need all of our blood going to our muscles. So we're going to constrict blood vessels within our core and send it to our arms and legs. We're going to dilate our pupils so we can take in as much information as possible. We're going to speed up our heart rate so we're delivering more oxygen to our muscles so they're better able to respond, things like that. And then parasympathetic is bringing them everything back to normal. So this is called rest and digest. Okay, so you would talk about impulses being sent. Like this is just referring to direction the impulses are going and where they're going to when we say the divisions of the nervous system. So I just kind of talked about this. Fight or flight, find ourselves in threatening situations, or maybe when you sit down next Friday for your test and you get very nervous. You're going to feel a little butterflies in your stomach, and what that is that blood vessel constricting in your stomach. So I talked about all of these: rapid breathing, prickly scalp, cold, sweaty hands, and then the autonomic, bringing things back to normal, conserving energy, and elimination of feces and urine. So more of the digestive processes. Just kind of going fast. So I said the nervous system is responsible for sensory input, integration, and motor output. So with that, this creates a reflex arc. So here you have a sensory cell. Sensory cells are typically unipolar. See how it only has one extension connected to it. So that's unipolar. Its ends, so the dendrites receiving information are called receptors. So these would be some kind of sensory receptor in the skin. Maybe it's a nociceptor. Nociceptors respond to pain. Could be a thermoreceptor responding to temperature. Anyways, sends a signal from the PNS, peripheral nervous system, to the CNS, central nervous system. In here, this is integration. This is your associative nerve or internerve, associative neuron or interneuron. They are multipolar. See how they have many extensions connected to them? So receiving signals from another cell, these are your dendrites. Dendro refers to tree-like, and then an axon coming out, and the axon branches to make many connections with many other cells. So this you only find in your central nervous system, in the spinal cord, and in the brain. Might be connecting, like this one is connecting the sensory and the motor, or it might be connecting between different interneurons, allowing communication to occur through the brain and the spinal cord. So the right talking to the left, or the dictionary talking to the Pictionary, talking to the parts that's going to tell your mouth how to say a word, right? So there's lots of interneurons connected, like when we are speaking even, which mine sometimes don't connect, I think. And then you're going to send motor output. Again, this is a motor neuron, and it's also multipolar, meaning it has many extensions. So the dendrites connecting with other axons, and then the axon sending it out. Okay, and we can talk a lot about the axon itself. It's covered with myelin sheath. That fatty insulation allows it to speed up transmission. Eventually, it connects with an effector. The effector is the muscle, organ, smooth muscle, skeletal muscle, or organ, gland, where it's going to cause a response. So it tells your salivary glands to release enzymes like salivary amylase. It tells your pancreas to release insulin. It tells your skeletal muscles to contract, yada, yada. Okay. I actually moved this slide to the next PowerPoint. So I thought you might want to break before I keep going. Do you guys want to take 5 minutes? Potty break or drink break or bathroom break, or I already said that one. Stretch break. Okay, so we're going to go ahead and take 5, and then we're going to move on with the action potential, where you'll see that reflex arc. So let me return to the Google World. New share. So this is the physiology. You have anatomy, which is action, and you have physiology, which is functions. So the nervous system functions through cell communications and action potentials and neurotransmitters. So why do we even need a nervous system? You can imagine to respond to environmental stimuli, like if you are a predator or prey, you need to be able to get away, you need to find your prey. So eating, finding a mate, the whole reproductive process, all of our body, look at, he got the bunny. All of our body processes. We need our nervous system to be fast. We need our nervous system to be accurate. We need to be able to reset it so we can go again. So that's what we're going to get into. So the nerve cell is called a neuron. So in the last, when you guys did your survey of the kingdoms, right, you identified neurons, which is a nerve cell. This section here is called the axon. So information, the arrows showing information transfer. So information comes in through these dendrites and goes through the cell body and goes out the axon. I was going to say something about the axon, and now I forgot what I was going to say. This is what it looks like under a microscope. Oh, if you bundle a group of axons together, that's a nerve. So you guys have heard of nerves, and this is a neuron, a single cell, whereas a nerve is many cells kind of wrapped together. The cell body contains the nucleus. It also contains the typical organelles like Golgi apparatus. You have ribosomes to make proteins, Golgis to package them and to modify them. And then you have I don't mean Golgi apparatus, I mean endoplasmic reticulum. The ribosomes are attached to the endoplasmic reticulum, so the ribosomes make the proteins, the endoplasmic reticulum transports the proteins, and then the Golgi apparatus packages them and modifies them before they are secreted for whatever purpose or used by the cell itself. In this case, we're probably talking about transport because we're talking about neurotransmitters that are going to send signals to other cells. So again, that big idea of structure meets function. There's many entry points, so you can monitor lots of environmental stimuli. There's only a one direction, so it's going to get where it needs to go. Just some fun facts. The longest neuron is in the white or in the blue whale, and the giraffe is the next, and then humans, 1 to 2 meters, like from your spinal cord to your toe or from your spinal cord to your finger. That's one long cell, right? So signal needs to move very far. So you have long transmission. So you have structure, medium, function. Neurons send signals out very, very fast. It's about a millisecond response time. When you think of signal transmission, you're thinking of a domino effect. So along the membrane of the cell, you have neurons, you have channels opening and closing. That's what you did in your pogo. So sodium was coming in and potassium was going out. And when one portion of the membrane, when sodium came in, it depolarized it, it changed its potential, it changed its charge, which caused the next channel to open. And so it's a domino effect in that sense, that when the inside becomes positive, the next channel opens. And then more sodium comes in, causing the next channel to open. And so that's what you were observing in that POGO. So there's a start, some kind of signal, some kind of change in the environment, whether it's internal or external, is going to cause an action potential. That first portion to depolarize, which then is propagated, meaning spread, the entire length of the cell. So if this was a neuron, it would go the entire length of the neuron. And then you need to reset, so put the dominoes back up, basically. And that would be to kick the sodium back out of the cells and bring the potassium back in. So this is your action potential that you were looking at. Notice the positive charges on the outside, the negative charges on the inside. So sodium is typically in a higher concentration outside of the cell than inside of the cell. When sodium comes in, as you see here, the inside becomes positive compared to the outside, because sodium is a positively charged ion, right? Which causes potassium channels to open. Potassium goes out of the cell, because these right now are moving. This is not the sodium potassium pump. These are moving with their concentration gradient from high to low. So then that causes the next channel to open. More sodium comes in, causing potassium channels to open. Potassium goes out, right? The reset is the sodium potassium pump, moving against the concentration gradient, kicking sodium out of the cell, pulling potassium back in. That's what you guys are used to. So the resting potential, typically outside of the cell, you have sodium, inside you have potassium. This is a gated channel. You can see the gate. So the protein embedded in the membrane is closed. When a ligand attaches to it, which is a signal chemical, then the channel will open. So for muscles, for example, acetylcholine is the signal, is the ligand that will bind to the sodium channel, causing the sodium to open. So for all of these kind of processes, we use examples in class, and it doesn't matter on the test what example they give you, they operate the same, right? So I'm giving you acetylcholine is going to cause sodium channels to open. They may give you dopamine. The process is the same. It's going to bind to a channel, causing the channel to open. All right, you don't need to memorize these charges, but maybe you need to read a graph, right? So typically our resting potential is at -70 millivolts. So you can actually measure this with its charge. So at this point, sodium's out, potassium's in, and there is no conduction taking place. So then the action potential is when the sodium rushes in, causing the inside to become positive. Right now, the potassium channel is closed, so potassium can't get it out. Once it's reached a threshold, once you've depolarized it, making it less different, once you've depolarized it enough, so maybe it's -50 millivolts, might be, and different organs have different thresholds, right? Your heart is going to be at a different number. So muscle cells, for example, maybe it's a -50. And that's going to cause the potassium channels to open. So at this point, we haven't yet reached it. Once you've reached that threshold, you've reached an action potential, and then it will spread, propagate. So the threshold stimulus here, you see -50. Once enough sodium has come in that we've now only have a difference of -50, the channels are going to open, and it's going to continue. Kind of like your fixed action pattern, right? Once it starts, it continues till its end. So this is just showing you sodium coming in, which causes potassium to open up, causes the next channel to come in. So these are voltage gated channels. So in this case, so the first one you needed a ligand, right? But these you need a change in charge, voltage gated channels. Propagation is spreading. So once one starts, the next one continues. And that's your domino effect. You can see the yellow part here, the yellow and red, this is your axon, the nerve ending itself. covering it, you have these myelin sheaths. So the myelin sheath is a fatty insulation, just like you put plastic or whatever it is, rubber on the outside of an electric cord, helps speed transmission because the propagation of the signal of the action potential can jump over this myelin. And so you only have to have action potentials occurring at these nodes of Renvier where the axon is showing. Okay. So that makes it go much faster. That's called sultatory conduction, from the word saltar, meaning to jump. So non-myelinated axons are quite slow. Many of our sensory receptors in the skin are non-myelinated. They don't have this covering. And so they have to have a constant action potential along the entire membrane, whereas saltar jumps and it's much quicker. So the repolarization occurs, sodium rushes in, we're depolarized, we're less different. Now potassium goes out. You're losing a positive charge, so you're reestablishing the original difference. So that's a repolarization. Usually there's an overshoot and right here, or an undershoot, I guess you would say, meaning more potassium flows out than you need. So that's what you see right there. And then you will reset using the sodium potassium pump, bringing it back to normal. So bringing it back to the original concentration gradient so that sodium can continue to flow out and potassium can continue to flow in. Because if you get rid of the gradient, they're no longer going to move, right? We don't want it to be at equilibrium. So the sodium potassium pump is the reset. Remember, 3 sodiums go out, 2 potassiums come in. And you have that coupled reaction, right? So it changes on both sides. So we've talked about that before. The refractory period right here, this is where you have your undershoot, too much potassium has escaped. So no contraction or no signal transmission can occur during this refractory period. So you have to wait till that refractory is over in order to send the next signal. And that is going to happen with that sodium potassium pump. So you have your resting potential here, sodium outside, potassium inside. You have your initial opening of the sodium channels in number 2. So the sodium is coming in. You've reached your threshold. Now you have your depolarization. All your sodium channels are opening. And then repolarization, potassium is coming in. I'm sorry, potassium is going out. And usually it undershoots, and then repolarization with the sodium potassium pump. So that's how you would read one of these graphs. I've seen these graphs on the test. I already talked about this one, so I can kind of speed this up. Sorry. So this is just showing you the sultatory conduction. Multiple sclerosis is an example of a genetic disorder that affects your nervous system by deteriorating the myelin sheath, this outer covering here. So if you deteriorate the myelin sheath, signals are not being sent appropriately. So there's a loss of signal, and so the cells are no longer talking to each other. You should be familiar with the synapse and the synaptic gap or cleft. So the axons of one and the dendrite of the other. So if this were 2 nerve cells, which it is in this picture, one would release some neurotransmitter and it would cross the gap to the next neurotransmitter. And that allows for the signal to continue. This could be a muscle cell. This is releasing acetylcholine. It will bind with the muscle cell after it crosses the gap. The smaller the gap, the faster the response. So there's lots of cells. You actually grow dendrites through practice. So any of you who do music or art or sports, these dendrites are growing, causing the synapse to get smaller. And that's why you get quicker, faster, better at your activities from practice. Also happens with practice on questions for the test, right? It's causing those synapses to grow closer together. Where is that information found? With practice, you find it quicker. And you got 71 seconds a question to find that answer. So here's what's happening at the synapse. You have that action potential depolarizing, causing calcium channels to open. This is the nerve cell. Calcium channels open. These are vesicles that are carrying neurotransmitters. The red dots are their neurotransmitters. The calcium coming in causes, that's like the signal for these to go through exocytosis, fusing with the membrane, opening up and releasing their neurotransmitter into the gap. And then you can see these will serve as ligands on the receptor protein of the next cell. If we talked about, I don't remember what section it was, but we talked about different chemicals, allosteric interactions, competitive inhibitors. So if there were another chemical that was similar to, in this case, acetylcholine, and it bound here instead, would you be able to send a signal to that muscle? No, right? And we know that the AP is infamous with like disrupting a system. So that would be an example of a disruption. So the neurotransmitter typically binds then, serves as a ligand to open that gated channel. And then depending on what's going to happen on the next cell, it's either going to continue the signal transmission or it's going to cause response. The neurotransmitter, in this case, acetylcholine, is then degraded and reabsorbed, reused. So you have acetylcholine esterase that breaks down the acetylcholine so that you can reuse it. So you can refer to the presynaptic neuron and the postsynaptic neuron before and after the synapse. One is receiving the signal. The postsynaptic neuron, it might be causing something to stop. It might be causing something to begin, right? So it's not always causing something to happen. It might be stopping a process. And we talked about those. So here's some of your neurotransmitters you might encounter. Acetylcholine is related to skeletal muscles. Epinephrine, you've heard of. Fight or flight. It's the same as adrenaline. So this begins that speed up process, increases heart rate, increases blood pressure, things like that. Dopamine, you might have heard of dopamine. It's in your brain, affects your sleep, your mood, your attention and learning. So it's kind of like a stabilizing neurotransmitter. Lack of dopamine in the brain is associated with Parkinson's disease. Some of the medicine that we take react as though it's dopamine. Like if you have a low level of dopamine, you might have medicine that you use to bind to those receptors. Serotonin, also found in the brain, also affects all those same things. These can be the weak point because we were just talking about how other chemicals might mimic the effect of that neurotransmitter. Nitrous oxide, carbon monoxide, some of the drugs that you take, stimulants, depressants. These all can stimulate or simulate other neurotransmitters. Hallucinogenic drugs. These are medicines that we might take that are taking the place of serotonin, for example. Other poisons, we talked about those with competitive inhibition where it stopped it. This is showing you acetylcholine binding to acetylcholine esterase. So then that's going to break it down. So an inhibitor is a neurotoxin. So it binds to that same location and then acetylcholine esterase can't break down. So the contractions continue. So then that's bad because there's no stop in the contraction. They become tetanic, which would be like lockjaw. But lockjaw does that too. Okay, that covered a ton of information. We still have 10 minutes. What questions do you have about this?